Hello again. One of the if you're doing molecular genetics or cloning, you'll find that you cannot run away from doing electrophoresis, right? And this is what you do when you want to separate your DNA fragments. Okay. Now, electrophoresis, how is it done? Is basically all right. Now, basically, what you do is you have a gel, right? It's like your regular agar gel, and you try to run your fragments inside the gel. Now, these gels are made of, well, it's called pores. You know, there's a structure. If you look at the molecular structure of gels, it has a lot of small, tiny pores. It's called pores or holes, and this is where you try to move your DNA in. How do you move your DNA? Is you apply electric current. DNA is negative charge, so if you put positive charge, so your DNA will move to the, it will be pulled towards the positive charge. Now, why is it? How does it work to resolve DNA? Because your DNA will move through the pores. You'll find that longer fragments will take longer time to move, will be more difficult to move. Shorter fragments will be very quick to move, correct? I mean, it's simple logic. So that means if you have a mixture of DNA, right? Let's say, for example, if you have a long piece of DNA, you want to cut a small piece out, you put in your enzymes, it will cut that small piece out, right? And then you take this mixture and you run on a gel. If you run it long enough, you'll find that the whole of the DNA will be separated because this fragment that you have cut is a small fragment, so it runs quicker. And then whatever is left are the long fragments, and it travels much slower. And in the end, you get two bands. All right. So basically, this is what you get. You're right. This is a cross section. This is your container. And then you put in your buffer. Your buffer is your electrolyte that allows electricity to go through. You put your gel, right? Your gels will have wells where you can put in your samples, and then you apply. Okay, this is wrong. All right, this is wrong because this positive should be on this side. All right, because then DNA is negative charge. If it's like this, then your DNA is going to move to the left. All right. This is wrong. So see, positive will be here, positive is here, negative is there. So you put your DNA sample in that, it's called a well, right? And then you switch on electricity, your current will flow through, and then your DNA will be separated based on fragment. And from this one, you will see that the fastest moving fragment must be the smallest, and then followed by the larger fragments and so on. All right. But how do you know what the size of what are the size of these DNA molecules? Now, normally when you run a DNA, you run along another sample of known sizes, and these are called as the reference sizes. Is also called DNA ladder. You know, just because you have many different bands for different sets of fragments. So you can buy this commercial ladder. This is an example, I think. Yeah. This is a ladder, a lambda DNA that's been cut. So you know that that fragment is 2027, that one's 2322, and so on. And you run this alongside your sample so you can estimate the size of the band that you're looking for. All right, so this is electrophoresis. These are your gels again. I'll just enlarge this a bit. Right, you put your samples. Now, this is a vertical system. Normally, your gels can either be horizontal on a slab, right? You can run it on a gel, or sometimes if you do sequencing, you'll have vertical systems, right? So you run them, but the principle is the same. You run it, and then you view the fragment. In this case, how do you view your fragments? In this example, you actually dye your DNA with ethidium bromide. What does ethidium bromide is a carcinogen. It actually binds to it can cause mutation if if you're not careful. So normally when you want to stain your DNA, you all wear all the protective gloves and so on because it's a carcinogen. So you mix this chemical with your DNA, it will bind to your DNA. What ethidium bromide does is that it will fluoresce if you view under UV light. So you can see here, you get very pinkish color. So these are your DNA. You can use other systems to label your DNA and then you can view them. But DNA on its own, you'll not be able to see it. But so this is how you actually see your bands. Okay. Okay. So I'm going to do an overall view of your cloning again. I'll be looking at your initial slides, and then hopefully you can put these pieces together of information together to understand what cloning is about. All right, I'll do that in the next slide, in the video. Sorry.